My dear students, today I am discussing the paper 1 of first MBBS Physiology subject of Dr. YSR University of Health Sciences, which was conducted or held on 20 January 2023. The multiple questions of this paper, that is set D of this paper, are discussed in the present discussion. I have at the end of these, I have given the reference for the correct answer, which you can verify. Further, I have found at least three questions which are not clear or can be disputed or contested because their stem is not correct, or there are dual answers, or there are some other things which I will explain when I am discussing those questions, and I have summarized them in the end about the whatever. Now, I will not go into the details of those questions which are easier and easily available in the standard textbooks. And however, I have given the reference for that, and you can pause and read those references and the context in which I have given. With this thing, I will start my discussion now. Question number 1. The greatest fraction of water reabsorption takes place in option A, proximal convoluted tubule, B, distal convoluted tubule, C, cortical collecting duct, D, medullary collecting duct. Please note that more than 2/3 of the solutes, especially the sodium, is reabsorbed from the proximal convoluted tubule. All the glucose, amino acids, the bicarbonate, and other constituents, including the urea, are reabsorbed either actively or passively. And because of the solute reabsorption, the water goes by the osmotic drag. So therefore, the answer to this question is option A, proximal convoluted tubule, where more than 65% or more than 2/3 of the water is reabsorbed. That is, the 2/3 of the filtered load of the water is reabsorbed. You can find this thing in Guyton and Hall, textbook of medical physiology, or other books, page 349. It's an international edition. I will not mention these things. I will just say that I have referred to the international editions. The next question, question number 2. Aspaxia is due to the combination of hypoxia and A, reduced oxygen carrying capacity, B, hypercapnia, C, hypocapnia, D, acidosis. Aspaxia is a condition in which there is obstruction to the airways, or as happens in case of pneumonia, where there is no gaseous transfer is taking place, or as happens in a drowning, you can see the water is there, no gaseous exchange can take place. Thus, there would be oxygen lack, and also the carbon dioxide is not eliminated. So there is a buildup of the carbon dioxide that produces hypercapnia. Hence, the answer to this question is option B, hypercapnia. You can find it in Genong's Review of Medical Physiology, page 634. Going to the question number 3, the cause for the characteristic features seen when a diver suddenly comes from a deep sea to the surface. Option A, increased arterial PCO2, increased pH, nitrogen bubbles in the blood, oxygen bubbles in the blood. Please note that the nitrogen is a fat soluble and easily dissolved. Under pressure in the deep sea, when a diver goes into the deep sea, under pressure, nitrogen gets dissolved in the fatty tissue or in the various membranes. When he comes out to the surface, or when he comes to the surface, there is a sudden drift in the pressure change, and this pressure change releases the gases which are dissolved. Just like what happens when you open up the carbonated bottle. So the nitrogen gases will come out. So the quantity of nitrogen is 80% of the total volume of the gases. Thus, that makes a greater difference. Even though the carbon dioxide is also soluble, but the quantity is only 0.03 or even lesser. So the answer to this question is the nitrogen bubbles in the blood. Option C, nitrogen bubbles in the blood. You have the reference listed here, the Guyton and Hall textbook of medical physiology, page 563. You can refer it. Then moving on to the question number 4. Ideal site for evaluation of cyanosis in a dark complexion individuals is lips, nail beds, tongue, earlobes. This question has a problem because the cyanosis can be peripheral and cyanosis can be central. If this question is, if the stem of this question is ideal site for evaluation of a central cyanosis in dark complexion individual, then the answer should be tongue or the mucous membrane. So since the stem is not clear, I would like that our students should take the benefit of the doubt, because ideal site for evaluation of cyanosis may be peripheral, may be central. So skin color does not affect the mucous membrane. And the lips, nail beds, and earlobes are less reliable because their color, if the colored complexion person is there, they cannot be detected easily, and there may be a different hue of the color. So this question is vague. It is true for the central cyanosis, but not for the peripheral cyanosis. Thus, it is disputed. It's not a clear question. Question number 5. The common type of electrolyte imbalance seen in blood transfusion is A, hyperkalemia, B, hypokalemia, C, hypercalcemia, D, hypernatremia. The answer to this question is A, hyperkalemia, because RBCs leak potassium during the storage, because when it is in a cold storage, they keep it in a cold storage, the potassium comes out. And this potassium is in the plasma of the blood which is being transfused. So when it is transfused, that will add to the potassium concentration of the recipient. So therefore, when we have to give this the transfusion in neonates and the patients with renal failure, we have to be very careful. So it is answer to this question is hyperkalemia. I've taken it from Harrison's textbook of internal medicine. And also it is available in Hindu Kurana, page 34. Question number 6. Cryoprecipitate consists of, I could search in Guyton and Genong. It is not there in the Guyton and Genong, and it is there in some in the Kurana. Cryoprecipitate consists of, answer A, packed cells, the platelets, the coagulation factor 1 and 8, coagulation factor 2 and 9. Cryoprecipitate is a source of fibrinogen factor 8 and 1 Willebrand factor, and even other factors like fibrinogen. So the answer is coagulation factor 1 and 8. So this is mentioned in Harrison's textbook. Also, it is present in Hindu Kurana, page 234, 2015 edition. The question number 7. Decreased arterial PO2 with a normal oxygen carrying capacity of a blood and rate of blood flow to tissue is A, hypoxic hypoxia, B, anemic hypoxia, C, histotoxic hypoxia, D, stagnant hypoxia. You consider here, in case of hypoxia, the oxygen carrying capacity of the blood is not altered. Oxygen carrying capacity of the blood remains same. However, there is no oxygen in the atmosphere or in the surrounding to be transferred. That's the question. As it happens in case of a COPD or in high altitude or any other conditions. So the oxygen carrying capacity of the blood is not altered, but the oxygen content surrounding is less. The alveoli do not get enough oxygen. And also the blood rate is not altered. So hypoxia may be a correct answer. If you have ticked hypoxia, I think you are right. Anemic hypoxia, hemoglobin is less. Oxygen carrying capacity is less. So it's not the correct answer. Histotoxic hypoxia, in the histotoxic hypoxia, you have oxygen capacity is normal. Okay, it is fine. And also the circulation is normal. However, the cells are not able to take up the oxygen because of the toxicity, maybe as it happens in case of the cyanide poisoning. So the answer C is also correct. So then stagnant hypoxia, yes, in case of a stagnant hypoxia, oxygen capacity may be normal, but there is a decreased flow rate. So answer to this question is, there are two answers. Hypoxic hypoxia may be right. Histotoxic hypoxia may be right. Therefore, you have to decide what you have done, and whatever you have done, if you want to have the benefit, you have to go further. So I have given the references here. You can just see that histotoxic hypoxia, it is there in Genong 23rd edition, page 617. In hypoxic hypoxia, you can find it in Indukrana, textbook of physiology, page 212. Okay, so this is a disputed question. Question number 4 was a disputed question. Question number 7 is a disputed question. Now, going on to the next question, question number 8. Lymphatic system exists in all organs except liver, B, lungs, C, central nervous system, D, intestine. Liver, lungs, and intestine are drained by the lymphatic system, but not the central nervous system. Answer to this question is the central nervous system. You can find this thing in Guyton, page 201. I am not reading whatever the contents, and I will leave it to you to read and analyze. Going on to the next question, question number 9. Celluloplasmin is responsible for the transport of iron, steroids, hemoglobin, copper. Celluloplasmin is a plasma protein which carries copper. So the answer to this question is D, copper, Genong 532. Going on to the next question, question number 10. Which of the following is not present in a normal urine? A, sodium, B, glucose, C, urea, D, creatine. In normal urine, sodium is present, urea is present, creatinine is present. The mistake is creatine, it should be creatinine. What is not present? Because in normal person, the glucose is completely reabsorbed. There is no glucose in the urine. The answer to this question is option B, glucose. You can find this thing in Genong's textbook of physiology, page 640. There is a table there. Then question number 11. Erythropoietin secretion is inhibited by. Inhibition is an active process. Somebody should stop the secretion of erythropoietin. Okay, let us examine this question. Hypoxia. Hypoxia stimulates the erythropoietin secretion from the juxtaglomerular apparatus. Androgen stimulate the erythropoietin secretion from the JGA. Products of RBC destruction, they will stimulate the erythropoietin secretion. Estrogen do not stimulate. Please understand, it does not stimulate. It is neutral. It does not stimulate. If estrogen, if the examiner thinks that estrogen inhibits, it is incorrect. Estrogen does not stimulate. Estrogen is a neutral thing. If estrogen is inhibiting, then all the females in the world should be suffering from anemia because of the erythropoietin deficiency. So the answer to this question, though it is estrogen, but it is disputed. It's not a correct response. The stem should be erythropoietin secretion is not stimulated by. That is the correct stem. Not inhibited. Inhibition is an active process, and inhibition is not the correct part of the stem. Therefore, this question can be contested. Here I read the thing which I have taken from Genong. Recent evidence suggests that the oxygen sensor regulation, erythropoietin secretion in the kidneys and liver is a heme protein that is coming from the erythrocyte destruction, that in the deoxy and stimulates the oxy and inhibits the transcription of the erythropoietin gene to form erythropoietin mRNA. Secretion of a hormone is also facilitated by the alkalosis that develops in high altitude. So now, this one is stimulated by androgens and hypoxia and the cell destruction things. Okay, erythropoietin secretion is facing catecholamines and the beta-adrenergic mechanisms. Okay, the answer to this question number 11 is C is not a correct response because the stem is not correct. It can be disputed or contested. Question number 12. Where does the first step in the production of urine begins? The first step in the production of the production of the urine is filtration. The filtration begins in the glomerulus. Then filtered things is collected in the funnel of Bowman's capsule. Then it comes to the proximal convertibule and subsequent tubes. So the answer to this question is the glomerulus, where the filtration takes place. This is the correct response. You can see it in the Genong's textbook, page 640. Afferent cranial nerves for deglutation reflex are A, 5, cranial nerve 5, 9, 10. Cranial nerve 5, B, cranial nerve 5, 7, 9. C, cranial nerve 7, 9, and 10. D, cranial nerve 8, 9, and 10. Look here, 8 is a vestibulocochlear nerve. So it's nothing to do with the deglutation. 7th is a motor nerve. It's nothing to do with the afferents. So the C and D are out. Now comes the real thing. When the food is after mastication, it is taken up in the mouth. In the mouth, the trigeminal nerve, the afferents present in the trigeminal nerve, they start the process of deglutation. The tongue goes up and presses against the soft palate and then hard palate, and then it moves the food bolus or food particles. So the correct answer to this is trigeminal nerve, 5th nerve, glossopharyngeal nerve, that is pharynx, and 10th vagus. If you have a problem in a glossopharyngeal nerve, you have a difficulty in swallowing. You have a problem in a trigeminal nerve, you have a difficulty in swallowing. You have a problem in the 10th cranial nerve, you have a regurgitation because it comes back through the nose, the new regurgitation. So swallowing is a reflex response that is triggered by afferent impulses generated in the trigeminal, glossopharyngeal, and the vagus nerves. It is present in Genong's page 471. Going to question number 14. Agglutinogen is present in the, very easy question. Option A, red blood cells, B, plasma, C, platelets, D, white blood cells. So agglutinogens are antigens. Antigens are present on the surface of the red blood cells, on the membrane of the red blood cells. Hence, the answer to this question is option A, red blood cells. Now, moving on to question number 15. Three phases of deglutation are a very easy question. Now, we have cephalic, gastric, intestinal, A. B is cephalic, pharyngeal, esophageal. C, oral, pharyngeal, intestine. D, oral, pharyngeal, esophageal. The correct response, because it starts in the mouth, that is oral mechanism. Then the pharynx takes over. Then the peristaltic waves generate in the esophagus. So that means the correct response to this question is D, oral, pharyngeal, and esophageal. You can have this thing in Genong's review, also in the Kuramas. The main function of albumin is determination of a blood viscosity, determination of osmotic pressure of blood, formation of a blood clot, defense. Defense is immunoglobulins, formation of a blood clot is a fibrinogen and other plasma proteins, clotting factors. Then blood viscosity is not by the albumin, maybe other heavy molecular proteins. Determination of it, the answer is because the albumin is nearly 3.5 to 5 grams or 5.5 grams per deciliter out of the total plasma proteins present. It would be having a greater attraction for water. It is present in the intravascular compartment and it retains water. It is the answer is determination of the osmotic pressure. To find this thing in Genong's review of medical physiology, page 482 as well as 532. So, well, question number 17. In a jugular venous pressure record, C wave is due to. Jugular venous pressure record, C wave, because C wave is due to. So when you record the jugular venous pressure or a jugular venous pulse, there are 3 positive waves, A, C, V, and 2 negative waves, X into Y. So now, let us see, options are A, atrial contractions, B, bulging of a tricuspid valve in the right atrium, C, venous blood flow into the atria, or D, jugular vein constriction. The answer is B, bulging of a tricuspid valve into the atrium, because the atrium, again, it comes back and that makes the C wave. So now, here I have given the details, which I have taken it from Genong's review of physiology regarding the formation of the ACV wave. Broadly, I will say that A wave is due to atrial systole. C wave is the transmitted manifestation of the rise of the atrial pressure produced by the bulging of the tricuspid valve into the atria during isovolumetric ventricular contraction. And the V wave mirrors the rise of the atrial pressure before the tricuspid valve opens during diastole. This is, I have taken it from Genong's page 512. Going on to the next question, question number 18. The true statement with regard to second heart sound is, it coincides with the R wave of ECG. It is due to the closure of semilunar valves. C, it is due to the closure of AV valves. It marks the onset of a ventricular system. You can just rule out these possibilities, the R wave corresponds with the closure of AV valves, the first heart sound. So the AV valves, closure of the AV valve produces the first heart sound. So the A and C are not answer for this. And it's not the ventricular system. So it is exactly the closure of semilunar valves. That means the pulmonary and aortic valves. That produces the second heart sound. The answer to this question is, it is due to the closure of semilunar valve, option B. You can find this thing in any books. I have referenced here guide. Question number 19. Fourth heart sound is due to closure of aortic and pulmonary valves. A. B. Vibrations of the ventricular valve by rush of blood. C. Ventricular filling during atrial systole. D. Closure of the mitral and tricuspid valve. Please note that usually we hear two sounds. Sometimes we may hear third heart sound. Fourth heart sound is recorded by the phonocardiogram. So now the answer to this question is, it is due to the vibrations of the ventricular valve by rush of blood. Closure of the aortic and pulmonary valve is a second heart sound. Ventricular filling during atrial sound may be third heart sound. Closure of the mitral and tricuspid valve is a first heart sound. So option B is the correct response, and you have this reference of Guyton here, and you can find it in page 284. Coming to the last question, very easy question. In a normal heart at rest, the stroke volume in ml is, one should easily get, it is 70 ml per min. So 70 to 80 ml is the correct response. It's not 30 to 40, or it is not 110 to 120 or 120 to 140, because there should be a limit. So the normal stroke volume at rest is 70 to 80 ml. Okay, this is, I have referenced 70 ml from each ventricle, but usually there is a range. So maybe 70 to 80 ml. You can take it even this side, that side, 65 to 875 or 85 or something like that. Okay, so now to summarize, following questions are not clear to me. Question number 4, stem is not correct. What is this question number 4? I will go back again. So question number 4, I will go back. Ideal site for evaluation of a cyanosis in a dark complexion individual. So here it should be central cyanosis. So the stem is not correct. Question number 7. Question number 7, the decreased arterial PO2, that means the oxygen carrying capacity of blood and the rate of blood flow are normal. So that can happen in hypoxic hypoxia as well as in histotoxic hypoxia. Hence, there are two responses. So now, then comes the erythropoietin secretion is inhibited. No, estrogen does not inhibit. Estrogen does not stimulate. It's a neutral thing. Hence, the stem is wrong. So now, to end up, I have just summarized them here in my last slide. Question number 4, stem is not correct. It should be central cyanosis. Question number 7, 2 correct responses, hypoxic anoxia or hypoxia and histotoxic hypoxia. Both are correct. Question number 11, no correct answer is available. Note that estrogen does not inhibit erythropoietin, rather it does not stimulate. Okay, thank you all. Please subscribe to the channel. Leave your comments in the box. Mention any topic or questions to be discussed. I want that after you receive the keys from the YSR University, please go through them with this YouTube lecture and then match what is happening, and you can always present to get your best markings. Good luck. Thank you once again.